Alright, this video is going to cover two objectives, Objective 4 and Objective 5 in Chapter 3. And they're going to be about two important processes that occur in the cell. And the first one is going to be DNA replication. And like the name kind of implies, we are going to be taking DNA and we're going to be replicating it or making a copy of it. And the purpose of DNA replication is to make two identical molecules of DNA so that when we divide our cells into two, each cell needs to receive a full set of genetic material. And so DNA replication is going to be a process that occurs during the cell cycle. That's going to be our last video for the chapter. And in the cell cycle, there is a phase known as the S phase, and the S stands for synthesis. And so this is the portion of the cell cycle when the DNA gets copied, and this has to happen before the cell divides, okay? So before we can make new cells, we have to make copies of our DNA, and that's what DNA replication is all about. So the basic process, and this is definitely basic, okay, we could spend a whole lesson or whole PowerPoint on just this topic here. I do want you to know that each step involves very specific enzymes. You can see some of the names up here. You don't have to know them for test purposes, but just know that enzymes are involved in each major step. And so in order to copy the DNA, the first thing we have to do is we have to open it up. So remember that DNA is two strands of nucleotides. The sugars and the phosphates are on the outside, and the nitrogen bases are on the inside. And so those nitrogen bases are held by hydrogen bonds, a weak bond between compounds. And so what happens is, in order to open it up, those hydrogen bonds between the bases have to be broken. So the first step is to unzip or open up the DNA molecule. Now, once that happens, you have those two original strands, and each original strand is going to be used as a template. Simply, it's going to be a guide for building the new strand, and it's going to be based on those bases, the base pair rule. So we have 4 nitrogen bases in DNA. We have A for adenine, T for thymine, C for cytosine, and G for guanine. And the way the DNA structure is formed is that the bases pair up. And A always pairs with a T, and a C always pairs with a G. And so that's how the new strands are going to be made. As the enzyme here kind of reads the original DNA strand, wherever it finds an A, it adds a thymine, and wherever it sees a C, it adds a guanine, and vice versa. And so the new strands are going to be built by an enzyme, and these new nucleotides are going to be added. Okay, notice the new strands are being formed in the inside here. And so once the DNA has been fully copied, one of those new strands is going to pair up with that original strand that it was made from. And then the other new strand is going to pair up with that original strand that it was made from. And we describe replication as semi-conservative. So each time a new molecule of DNA is made, there's an original strand and a new strand in your new DNA molecules. And by the end of DNA replication, we have 2 new identical DNA molecules, and that will be followed up by cell division to make 2 new cells. And so here we have the original, it gets opened up, the new strands get built based on those base pair rules, and in the end we have an old strand paired with a new strand. Alright, the second process we're going to talk about in the video is protein synthesis, and this is all about making those macromolecules that we call proteins. So just a quick recap, proteins are made up of amino acids, that's the monomer for proteins, and the proteins come in 4 different structures. So it starts by making a chain of amino acids like we see here, and then this chain will go on and twist and fold, and it's going to take on a very specific shape. And so the process of protein synthesis is just building this protein chain that we see here. Now, it starts with DNA because the instructions for building our proteins are in our DNA, which is why we call it our genetic instruction. And so we can kind of sum up the whole process of protein synthesis with this statement right here. DNA to RNA to protein. And so we're going to start in the nucleus where the DNA is found. And the problem is the DNA cannot leave the nucleus. The original blueprint is going to be stuck in the nucleus. as you can see, DNA has 2 strands, and so it's actually too big to fit through the nuclear pores. Now the issue is that the proteins are built at the ribosomes. And so we're going to have a middleman. Okay, so the instructions are in DNA, which is in the nucleus. We're going to build the protein over here at the ribosome. So what we need is a way to get the message from the DNA to the ribosome, and that's going to be the purpose of RNA. And so the overall process of protein synthesis is going to take place in 2 phases. Now, the first phase, transcription, is the first portion of that statement. It's taking the DNA and making a copy of it in the form of RNA. Now, this is kind of similar to DNA replication, but there's a couple of major differences. Number 1, we're only copying one piece of the DNA, what we call a gene. We don't need to copy the entire DNA sequence, and so we're actually only copying one side of the DNA molecule. The other big difference is this time we're making RNA, and so we're going to be adding RNA nucleotides to build the new strand. Now, when it's RNA, there's only going to be one strand. There's going to be no T's. It's going to have the sugar ribose. And so we're going to build it kind of like we did with DNA replication, but this time, anytime there would be a T in DNA, there's going to be a U in the RNA. So once again, it's going to be built based on those complements. Okay, this time it's A to U though. And so in transcription, we're just going to be copying that one piece of DNA to get the instructions that we need for building that one protein. Okay, and that's what we call our transcript. Now, because DNA is involved and DNA is stuck in the nucleus, transcription is going to take place in the nucleus. So now what's going to happen is we need to build that protein at the ribosome. So the RNA, in this case it's mRNA, or messenger, it's going to leave the nucleus and it's going to travel to a ribosome so that we can build that protein. And so at the ribosome, that mRNA, and this is very simplified, but that mRNA is going to be read 3 letters at a time. So remember that proteins are made up of amino acids. Now in our genetic code, 3 letters in that RNA are going to determine what the amino acid is. And so that mRNA is going to be read 3 letters at a time, and every 3 letters we're going to have an amino acid added until we get to the end. And so ultimately what we're going to be building is what's called a polypeptide. That's a basic protein. It's just an amino acid chain. Now once the polypeptide is made, it's going to go on and twist and fold and become that protein that's functional. But in protein synthesis, we're just building that amino acid chain. And it always goes back to the instructions in DNA. And so we start with the transcription in the nucleus. Okay, the DNA is going to be copied into an RNA transcript. We call that mRNA. And then the RNA is going to leave the nucleus. It's going to travel to a ribosome where it's read 3 letters at a time. And in the process of translation, we're going to build our amino acid chain here, which will then go on and form our protein. And that's all for this video.